Anybody want to try at the front? Try lah. Very easy. No, I think it's good that you train your brain, not for the sake of memorization, the sequential step. Because when you can memorize things in the right order, that actually a good mental exercise. Pretty much like this. Siapa nak jadi guinea pig? Macam aku je buat kan? Oh, by the way, when you look at the Z scheme here, I forgot to mention earlier this morning. Look at on the y-axis. Look at the unit there. What's that? What's that? What's M? Midpoint potential. Okay, the energy of midpoint potential in the unit of volts. You can see here that as the molecule get excited, in this case the chlorophyll, you can see that the action potential become more negative. This is important because in a bit you're going to learn something about charge separation. So the moment this become negative, why it becomes negative? Because one molecule, the ground state P680, the electron gets ejected out. Okay, so when this happens, a phenomenon is occurring that is known as charge separation, meaning that the Pheophytin here will accept electron. Remember, Pheophytin is a type of chlorophyll without magnesium in the middle. Chlorophyll that you learn, it has the magnesium in the middle. That's why it appears green to our eyes. Okay, but Pheophytin, everything looks the same, but there is no magnesium in the middle, right? So the color of Pheophytin, it's pretty much like when you brew the green leaf, the tea. You know, you lose the color, right? It turns brown, it turns gray. Those gray, those brown, Pheophytin. Magnesium that have left the chlorophyll molecule. Okay, so these become negative because it just gained electron, and these become negative or positive, and electron is ejected out. This guy become negative. What happened here? It becomes positive. Yeah, so one molecule positive, one molecule negative. So there are two charges here. So this is called charge separation. Okay, so because the negativity is more concentrated in this area, that's why you see the mid potential voltage becoming a bit more negative here. Okay, if you look at the Guinness Book of Record, this P680, P680 here, this is the strongest strongest oxidizing agent in biological kingdom. This, the P680 that has its electron ejected out, the whole P680. It is so hungry for electron to the point it oxidizes water. Can you simply remove hydrogen from oxygen in a water molecule? Take a knife. Can you do that? It's not going to work, right? But this thing, it is so electronegative to the point it can split open the water molecule so that oxygen is released, hydrogen proton is released as well. That's stronger. So this is very strong. I do not know any other places. I think this Guinness Book of Record hold this record for this. The strongest oxidizing agent in the biological kingdom. If this is the strongest biological oxidizing agent, this would be the strongest reducing agent for P700, because the whole thing is redox reaction. One guy is reducing, the other guy is oxidizing. Okay, right. I'll include here, but you can read later. This is how the experiment was done to understand this thing, the midpoint potential. This thing here that you saw earlier, the M here. Yep. So when a solution, such as the solution of a pigment, is bombarded with some kind of energy, if absorption and excitation of molecules happen, you will get this charge separation continually. And this charge separation, depending on the magnitude of it, can be detected by voltmeter. Right. That's how the battery works. The battery behind your remote control and so on. It's got the negative terminal and positive terminal. The negative part will actually travel throughout the remote before it return back to the positive side of the battery. Okay, so the stronger electrolyte content in the battery, the faster this is going to go, the more voltage that you're going to get. The regular battery that we have, what's the voltage of it? I think 1.5. 1.5, right? Compared to the Smart Tech battery, what's the voltage? You know Smart Tech? Yang the square, not square, rectangle battery. 9. Yeah, see? It's a lot bigger, it's a lot heavier, lots of electrolyte. So electronegativity is high. So you detect more voltage in there. Yeah. Think of it like the content of the battery in here. Okay. I'm not going to teach electronics here because people are going to cry further. Yeah. So you have seen this before. 4 proteins important for this thylakoid. What are those? PS2, PS1, cytochrome B6F, and also ATP synthase. Please remember that these are the only confirmed integral protein of thylakoid membranes. Other proteins are present, but they are not necessarily integral protein, meaning that protein that gets embedded in the membrane of thylakoid. This plastoquinone here, can we call it the integral protein? You can't really actually, because this guy is actually moving. It's blobbing around in the fluidity environment of the thylakoid membrane space in here. Okay? These, they are pretty much stay where they are. That's why they are not called carrier. Plastoquinone, all of these, plastocyanin, we call them carrier because they can actually move. You don't see it's moving here, but they are actually moving. Why they move? Because they have been excited by electron. They have accepted electron. Now, they need to wobble from left, meaning that they are facing the stroma side. Now, they face to the other side, to the luminal side of it. Okay? Right. Ooh, what's this? So this, in essence, is the story that I've been talking about earlier, how the movement of this electron. I think this is something that you can read on your own. But just to highlight something here, you're going to see this, where is it? Is it mentioned here about the Q cycle? Is it mentioned here? Anything about the Q cycle? Yes. I know it's there, but I should know this because I made this note right. First point, second point? Second point. Yes, the oxidation is a complex process called the Q cycle. Just now, what you saw here, the movement of electron going up, down, down. That's Z scheme. Now, there is another movement of electron that is called the Q cycle. Q cycle specifically happened here, involving plastoquinone and cytochrome B6F, which you're going to see in a big detail in the coming slides. Okay, so How come you get two products here? Remember, the end of the story here, you get two products. You get NADPH and also ATP. NADPH, you already know how that came about. ATP is actually the results of ATP synthase action. You see, as the electron moving along the Z scheme, it's not merely moving. Whatever that it touches along the way, all the protein, it will cause the protein to grab hydrogen proton from the stromal side, bring into the luminal side. That's why you see all this like a recycle symbol here. Yeah. So you can picture the plastoquinone now. It is a protein. It is moving. It has two side of faces. One face facing stroma, one face facing lumen. Then it has the capacity to capture. The moment it capture, it will start to wobble, turn the side of the body. When it turns the side of the body, not only empty turning, it will bring hydrogen from the stroma into the lumen. Remember, the splitting of water due to the oxidizing of P6H already producing hydrogen. Now, all this protein bringing further hydrogen into the lumen. At one point, you are going to get higher and higher and higher hydrogen concentration inside the lumen. Higher hydrogen, what happens? Increase or decrease in pH? Decrease, becomes more and more acidic. So tell me now, is that an equilibrium? So in stroma side and also the luminal side, there is an imbalance of pH because in the lumen, so much hydrogen. These hydrogen, they need to live because that's the law of universe. Equilibrium need to be reached somehow. However, they cannot just exit from where they came from. There's only one way to go out, through the tunnel, and this little tunnel we call ATP synthase. Yeah. So the smart design about this integral protein is that when the hydrogen rushing to get into this ATP synthase tunnel to live and go to the other side in order to reach equilibrium, this will cause ATP synthase. You see the rotor head here? So you got the body embedded in the thylakoid phospholipid layer, and this is the rotor head. This rotor head actually can turn like a turbine. You see, you got a dam, the dam coming with all the water in, and then the turbine will start to spin, right? Kind of like that. So this surge of hydrogen proton, the rush of hydrogen proton, will spin this ATP synthase rotor head. And guess what? This rotor head kind of look like a many pillow come together, right? So each subunit of this head holding two things: ADP, adenosine diphosphate, and also PI, inorganic phosphate. These two things are not merged together. They are individually separated, but being held loosely to the ATP synthase. But the moment hydrogen exiting this rotor head and the rotor head is spinning, the spinning of the rotor head will jam this ADP with inorganic phosphate together, then fuse them, kind of like laminating machine. Fuse them together, bam, you get your ATP. That's how you get ATP. And the same story actually happen in your cells as well, but not in chloroplast, in mitochondria. Okay, we have that as well. Okay, so that's how you get ATP, right? But you will see later that in photosynthesis, between ATP and NADPH, you actually need more ATP. Okay, so what's the difference between these two? They look like they are something to do with energy, right? ATP is to do phosphorylation. To do process of phosphorylation, meaning that adding phosphate to any molecule that it likes. Phosphorylation. You phosphorylate something. When you phosphorylate something, meaning that you energize something to become something else. Okay? And commonly, that's why ATP is called energy currency in the cell. For NADPH, it is called as the reducing agent, reducing molecule. In some book, they are called electron donor. Okay, meaning that it's not energizing the molecule, but it donates its own hydrogen to the molecule so that the molecule changes from the oxidized phase into a reduced phase. Pretty much like this thing here, plastoquinone that you saw in the morning. Plastoquinone, nothing attached to it. The moment it receives one electron, it becomes plastosemiquinone. The moment it receives two hydrogen, meaning that somebody else has donated hydrogen to it, now it becomes plastoquinol, or the full name of it is plastohydroquinone. Right? ATP is not involved in here. This is the work of NADPH, reducing agent. Okay? Right. Yeah. So, yeah, that's the story for this final point. Okay? So, remember again, you have your thylakoid membrane. Your thylakoid membrane got two regions. What's the middle region here? What's it called? Tak sampai 3 hari. Grana lamella ke lamelli? Lamelli. Plural. Banyak ni filament. Grana lamelli. What about this one? Stroma lamelli. Okay, please remember that. Right. Okay. I put a little note here to help you with this explanation. Okay. What is hydrogen atom? What is hydride? What is proton? Okay. Some in many biology book, Campbell or anything, you're going to see that they talk about proton motive force. That is actually hydrogen. Positive charge hydrogen moving about in chemiosmosis. They don't call hydrogen, but it's actually hydrogen. They call it proton because this is the form, H plus. If it's H minus, this is the hydride of it. If it's no H, no symbol, no negative, no positive, that's actually hydrogen atom. Right? Okay. Yeah, so I put a little note here for you to revise what is meant by carrier, what is meant by acceptor. Right? I've mentioned that in the morning. Ooh, what's this? You see, I think most of you maybe have not learned this, but if you take electronics, you're engineer, you take electronics, you learn this. So because in electronics, you need to learn something about photo-induced electronic components. So these photo-induced components in electronics, they too undergo electron excitation. And that's why you see this symbol here. So the symbol here is actually to show the state of chlorophyll, ground state of chlorophyll, and then excited state of chlorophyll. In the ground state of chlorophyll, it has these properties: poor oxidizing agent, poor reducing agent. Your friend, when she is not laughing, no emotion whatsoever, right? Because you have not started to tickle her. But the moment you tickle her, or in this case, you give light, the electron here, Y2, that's the duplet configuration, remember? Either octet or duplet. So this is duplet. So the moment light comes in, light of correct energy, correct nanometer, not just any light. In our case, for chlorophyll, it's either 680 nanometer or 700 nanometer. What about bacteria? You saw just now. 870. That's in bacteria, especially purple bacteria. Okay. So let's say that you got your right kind of nanometer of light energy. This will be excited. You see, when it gets excited, the electron now, it does not leave the molecule just yet. It becomes excited first. It go to the orbital. Excited orbital. If I can put it in drawing, it's something like this. So you have your atom. Welcome to chemistry. So you got your nucleus in here. Nucleus. So you got your, let's say that this is the outermost orbit of it. And your electron, say it is that these, they don't stay in here all the time. Okay, sometimes they are here, sometimes they are there. You can think of these are actually connected to the nucleus with a rubber band. That's why they can vibrate, because rubber band, when you kind of, tie up your friend's leg, you can pull it. You see the rubber band vibrating? Kind of like that. This is ground state. The moment energy comes, suddenly an imaginary orbit start to happen. It is still the same electron. It's not like the electron suddenly got new electron values. No. This is temporary orbit. This guy will go up here. So this is what we call the excited orbital. However, bear in mind, this is only femtoseconds. Super fast, because it's not stable. Either you leave the orbit to go to a new molecule or come back to reality. It won't stay there very long. Okay, so that's why one of the things that you can do in agriculture is actually to increase photon capture and photon utilization. Okay, maybe some of you have learned this. We call it 5PS2, the efficiency of photosystem 2. Not only to capture light, but to actually utilize the light energy to be turned into chemical energy. Okay, so this guy, the moment it becomes excited this way, now it has two properties. It is a good reducing agent and also a good oxidizing agent. Why it is a good reducing agent? Because at this state, it can easily, what? It can easily donate because it's already out. It is, at the same time, this guy here, good oxidizing agent because this actually already a hole. It is super hungry. This is what I called it earlier, charge separated state. It is separated. I know I think most books do not have this kind of explanation. I saw it many years ago. Thanks to a professor at King's College. Right. All these universities, if you go to the UK, you go to all these countries, they have open lecture. Right. Malaysia do that. They can open lecture, but nobody come. They do open lecture. Even the barista at Coffee Costa Bean can come to listen to the lecture about anything. At one point, the lecture that week was about this kind of thing, and I attended. Kind of like, oh, interesting. Oh, by the way, they use the blackboard. Macam cerita apa? Oppenheimer. Right. So, these professors, they are very fluent, very eloquent with their explanation. Okay. So that's why you need to have the right people in life to talk about the right thing. When you are still young, go to listen all of these things. If I didn't attend those things, I wouldn't be able to know about this. I mean, this thing is around in the internet, but I wouldn't know. This is the right way to explain it. Right. Okay. So that's the explanation for this diagram. Ooh, what's that? Oh yeah, charge separated state. I already mentioned that. This thing here, the coming back to reality here, it gets excited, then it comes back. I think there's a terminology for that. Oh, this thing. Recombination process. Electron can return to the reaction center and reform the ground state with the loss of the stored energy. This wasteful recombination process, this thing, it gets excited, but it's not proceeding to the next molecule to do electron transport chain. Coming back to the ground state, wasteful process, we call it recombination process. Yeah. If the plant is very efficient, very productive, this is not happening so much. Okay. Remember from last week, there are 5 fates of excited electron. You can open your note from last week. It should be on page, what page was it? I know it's somewhere. How many fates of excited electron? 5. What are those? Yep, this slide here. All this while in my classroom, I only taught 3 states of energy when chlorophyll get excited. It's either photochemistry, heat, or fluorescence. Now you got 2. What's the other 2? The other 2 is actually either the energy is used to form a triplet chlorophyll, the 3 chlorophyll star, or it can actually get passed to other molecule and come back to itself. Okay, so these are, that's why it's 5. So you can read further, okay, to recall. Don't let the memory to go far away. Call it back. Okay. So this basically, this story is reiterating all this lecture from last week. Okay. Yeah. So good luck with reading that. Yeah. Now, talk about the reaction center. Reaction center chlorophyll of 2 photosystems absorb at different wavelength. 2 terminologies you need to know here. Number 1 is absorption spectrum. The other is action spectrum. They are different, okay? Absorption spectrum is whatever the pigment is able to absorb maximally. For example, in our case here, a chlorophyll, we know it can do the absorption around the blue region, around 470, and then it absorbs maximally again around 680 or 70, something like that. And it is not absorbing anything in between the yellow, the orange, the green. These get reflected back. That's why leaf appear green to our eye, because chlorophyll only absorb 2 colors predominantly, blue and red. Okay. What about the action spectrum? Action spectrum, it is still talking about the absorption by the pigments, but what happens as the result of absorption? They quantify it further. Just now, the ability to absorb. Now, the ability to utilize. So we want to quantify the utilization. So in the graph here, it is utilized in the form of oxygen evolution, the release of oxygen, meaning that the chlorophyll has absorbed the energy. When it gets excited, what happened to the chlorophyll? What happened to the electron? It stays there or it gets ejected out? It gets ejected out, right? Go out. Now the chlorophyll got a hole, and this hole is a strong oxidizing agent. Somebody needs to fill in the hole. Who's filling in the hole now? Water. Remember the Z scheme just now? This thing. There is a name for this actually. It's called photolysis. What's my pen? Photolysis. The split of water, H2O, you got H plus, you got O. Photolysis. Okay? Yeah. Yeah. So you can measure this, and as you can see here, the action spectrum is not entirely the same as the absorption spectrum, even though we are talking about the same chlorophyll extract here. The action spectrum is somewhat slightly lower because this is biology. Biology, from time to time, you have inefficiency because some of the energy is dissipated to the side, glow or anything in the form of heat. So this heat is not oxygen, cannot be quantified. That's why it's a bit lower. Okay, but this is only happening in the blue region. In the red region, which is lower energy, the chlorophyll can mostly deal with this energy, absorb and utilize, absorb and utilize. Okay, coming back to here, the excited states, there are two actually. This is also from last week. If you open your notes, this figure here, there are two excited states. It's called higher excited state and lower excited state. If you are dealing with blue light, you are going to go higher. Maybe somewhere here. Oops, it's a bit more. It is still in the region of excited orbital, but have not left the current molecule. Okay, that's why it needs to immediately come back to the lower excited state, which is achieved by the red region here. Okay? Yep. So which one is going to do photosynthesis? The red region. Okay? The lower excited state. Okay? Because plants, when they are outside here, it looks very scorching hot out in outside there. They can't control the amount of energy coming from the sun, cosmic ray and all. Okay, so more than often they have to deal with this higher excited state. But plants have pigments surrounding reaction center. These pigments act like sunscreen. You know, you apply sunscreen, go to beach, right? Plant got sunscreen as well. What are the sunscreen? Carotenoids, xanthine, chlorophyll B. All these are the pigments to filter or dampen the high energy effect coming from the sun. Yeah. Ooh, what's this? Photosystem II reaction center is a multi-subunit. Oh, okay. This thing, you need to play. You know that there's a topic in modern math, in SPM math. You deal with the drafting of shape, and then you need to look at the 3D way of it. Sekarang tak belajar. Kind of like geometry, because I remember in my class I was the only one yang jawab soalan tu because it's optional. The rest they kind of jawab soalan susah lah, log benda tu. I didn't answer that. I answer soalan macam ni. Malas nak kira. Okay, so this is actually photosystem 2, meaning that it is a protein. To show what is inside the photosystem 2 protein, what's inside it? So inside it is what we call as multi-subunit, pigment plus protein complex. It is never one thing. The protein actually contains various pigment attached to it, come to many parts, turn everything into one complex. Okay, in A, I can show you here. So that's the enlarged version of it. So A is the when you look at the reaction center from the top. Here is your reaction center. Here is your photosystem protein. I can use this. Here is your reaction center protein. It is embedded in the membrane. two double layer membrane, it's in here. Okay, so the first image is actually looking up at this, the top of it. Yeah, so you can see that there are protein dimer, D1, D2, and also some other protein as well, CP43, CP47, okay, and they are actually connecting to the pigments, okay, meaning that the pigments never stay alone because somebody needs to anchor the entire complex to the membrane of thylakoid. Remember, when this thing is dealing with the super sunlight, everything will get very hot. Somebody needs to move, somebody needs to stay in place, somebody need to change color, somebody need to eject electron. There's like a busy mini city. Okay, so when it's a busy mini city, so they need the whole complex to have an anchor, to have a loose part, to have a fixed part, and to have a modifiable part, so that the whole thing can work just smoothly, right? Okay, so that's how you look at it from the top. This is from the cross section, from the front of it. First one, you look from the top. This one, you look from the front of it. So from the front, this is how it looks like. You see all these orange, I think these are the pigments, should be the pigments. View from the luminal side of the supercomplex include additional antenna complex. Remember, antenna complexes are the pigments. What are the pigments? Carotenoids, chlorophyll B, chlorophyll A, xanthine, viola xanthine, so on. Anthocyanin, whatever color that you can think of, depending on the plant species. Okay. So in extrinsic oxygen evolving complex. So this thing here that you can see, orange and yellow ovals here, these are called as oxygen evolving complex. Usually these things, they have manganese attached to it. The process of photolysis requires manganese. Okay, meaning that their function is not to give color, not to eject out oxygen, but rather to facilitate in the process of removing hydrogen and oxygen in a water molecule. Remember, it's not easy to remove H2O, okay? You want to separate this, try use knife all day long. It's not gonna happen. So these complexes, they are designed to do just that, remove, okay? Right, and finally, this is from the side of it. If this one is the look from the front, this side look from that. Oh, kau nampak perut dia ke depan. Macam dia, apa kau gelak? Because you got one, right? So this is the body. This is the protruding tummy of it. And these are the oxygen evolving complex. You can see that they themselves don't like to be associated inside. They are actually protruding out. Because why they are protruding out? This side actually facing lumen. They need to immediately release the oxygen into the luminal side of the thylakoid. And these are all not in the form of gas, okay? You are in water right now. All of this. So oxygen that get released is a dissolved oxygen, okay? Dissolved or gaseous oxygen, still oxygen. So nobody is allowed to play with lighter in here. Right, okay. And this is a structure of photosystem as well, but the example is taken from bacteria. This one is from plant. So the purpose of having this diagram is to show how similar plant-based photosystem protein versus bacterial-based photosystem protein. It's pretty much look the same actually. You got the stroma side of it, the lumen side of it, you got the D1, D2 protein. Are we the same organism? How come? How can they got the same? This is two different things. You're talking about one about bacteria, one you're talking about your citrus tree. Completely different organism, but you go to this level, they are actually made out of the same thing. Yeah, in fact, they got the heme as well. We too got heme in our hemoglobin, right? So that's why all these, crazy people, they come up with the evolution theory. We all come from the same cell. Yeah. So they are basing on this actually. Just because you are sharing something similar with somebody else doesn't mean that you are sharing the same akong with that guy, right? You got leg. The ghost also got leg. No, ghosts don't have leg. Apa ghost ada? Ghost got head, you got head. So you want to tell people you share the same ancestor with the ghost? No, it just happened. That's the best design the universe has decided for our situation here. All right. That's the reason why the planets in the form of sphere. Why one planet suddenly you have Mercury? No. I want to look funky today. Nah. Let's go oval. So suddenly you got Mercury in the shape of egg. And why can't Mercury look like eggs? Why all the planets look in the form of sphere? So there must be a force behind it that dictates for throughout this universe, even though you are of different entities, this is the design to be used. Okay, so that's why you keep on saying this, because I don't want to go into the evolution a rabbit hole. Okay, there's a long discussion that nobody going to win. Right. Okay. And this is the C. C is the see, it's contain magnesium here. Magnesium. Not magnesium, manganese. Manganese containing water splitting complex. This thing. I told you earlier, right? It contains the manganese. Yes, it shows here. It contains manganese here, right? And also calcium, right? So this is something. Don't worry, nothing in the exam going to ask this, but to give appreciation, when you look at the bacteria later, don't think that bacteria, just because they look small, they don't have what you have, right? Okay, right. Okay, we have talked about this. The water is oxidized to oxygen by photosystem. So I've got 15 minutes left. Yeah. So be familiarized with these terminologies that I have explained. Oxygen evolving complex. Okay, the tummy guy here for the vascular plant. This is picture for vascular plant. This is picture for bacteria, right? Yep. So actually, you don't have to worry about this. This is about the excitation states. I think if you learn chemistry in Depot, you're going to learn about this. The S states of this thing. The water is split, it becomes more and more oxidized due to the absorption of energy. Pretty much like this thing. It has different state of negativity depending on how many extra or less electrons attached to it. It's not like it's not functioning, but it's the question of stability now. Yeah. If you have, because our body, the way it works is, this thing needs to be present at a specific rate or ratio. If you have too much of plastohydroquinone, you will exhaust the pool of recycling substance. So that's not the best way to ensure continuous energy supply throughout the day, because when you are sleeping, because this thing is happening in our body as well. It's not plastoquinol, it's just ubiquinol. In our body, it's called ubiquinol. It's called ubiquinone. Plasto replaced with ubi. So that's the thing present in us human. So this is just to say the S state or the oxidizing state of it. Oh, I already explained this, right? From plastoquinone, how did it become? Yeah, nak aku cerita awal kan, pada ada dalam slide. Lupa. So you got plastoquinone. Remember what is plastoquinone? I'm going to ask this again. What is plastoquinone? What is plastoquinone? Two words. Electron carrier. Electron carrier. Why it cannot be electron acceptor? Is it electron acceptor or electron carrier? Carrier. Why? It accept and it bring to somebody somewhere else. It move the electron. Okay. NADP, electron acceptor, because it just use the electron just for itself, not going to give to somebody else. So plastoquinone, the oxidized form of it, the moment it receives one electron, see, plus one electron here, it becomes plastosemiquinone. And that's how you write it, PQ dot with the negative sign to it. Then when you add one more electron to it, with the presence of two hydrogen, yeah, I want to talk about this. You see here, we know that this electron comes from the ejection of chlorophyll molecule before it, right? Where this came from? This from the stroma. Remember, I was telling you about the plastoquinone got two phases, the stromal phases, luminal phases. When the body has accumulated two, meaning that two rounds of excitation, it will automatically grab two hydrogen proton from the stroma, attached to it, the body, now become plastohydroquinone, PQH2. In fact, this thing has been commercialized now. They turn it into human supplement. PQQ. Did I told you about that, to increase mitochondria number. PQQ. Yeah, PQQ is actually this thing, but in human form. If you eat this, the effects to human body is you will increase mitochondria number. Yeah, they call it PQQ. So this is in the plant form, of course. Yeah, don't eat just yet, unless you are an athlete. Okay, right. So yeah, I think there's two more. Then we can stop about the cytochrome, and then about the PS1 protein. Yeah. So electron flow. Electron, after the plastoquinone has captured the electron, now it needs to pass to the, my marker. I need to have it written some. Is this permanent? Oh, habislah. Anybody nak lukis? How do you say this? Ground state, P6S0. Ni? Excited state. Bukan happy state. So, first molecule, siapa yang terima? Pheophytin. Then, who receive? PQA. PQB. Lepas tu? Cytochrome lah. Cytochrome B6F. So we're going to talk about this now. Yeah, we're going to talk about this. So where exactly is this cytochrome B6F? It's somewhere in here. It's actually it's rather a big thing. So this cytochrome B6F, if I want to enlarge it, it will become something like this. It's actually a complex on its own right. So PQ, then become this. So there is a site here, it's called Q0. Okay, so Q0. Cytochrome F. If it goes up, cytochrome B lower. Cytochrome B higher. Dekat sini boleh jadi PQ tu. And this is called QI. Tak ada ke slide nak tolong aku mengajar? Kenapa aku guna? Oh ada. Oh, penat lagi. Pada tadi lagi. Macam itulah. What you need to understand is the moment just before the electron gets into cytochrome B6F complex, which is another type of integral protein, there's a bunch of things can happen. Okay, number one, the electron will first attach to the initial site called Q naught. Okay, this is the first site inside the cytochrome B6F. And there are two paths this electron can go about. It can either, number one, go down way this way. Okay, oh, I forgot this thing. This iron, sulphur, what's that? This R is the name of a scientist actually. Iron, sulphur, risk or rest? I think risk, iron, sulphur, protein. This thing, FESR, that thing here. Then it will be transferred to cytochrome F. So the electron can go down here and then will be passed to plastocyanin, another electron carrier, then proceed straight to P700. If it follows this is the regular Z scheme. Okay, nothing special, just a regular Z scheme. However, this electron can also go up. Actually, if you think about it, it can happen simultaneously because this is very fast coming here. One can go down here, one can go up here. If it goes up, it will go to the first heme group called cytochrome B. L means lower state. H means the higher stage of it. And then it will be passed to this free random plastoquinone. If it do it one time, the plastoquinone from plastoquinone will become plastosemiquinone. That's why you got it here. Yeah. The second round of it, if it happens 2 times, this plastosemiquinone will absorb another 2 electrons, then it will start to get hydrogen. So the key is plastosemiquinone need to accumulate 2 electrons before it decides to grab hydrogen from the stroma and change itself into plastohydroquinone. Yep. Yeah. And the site here, the exit site here, it's called QI. I don't know why is it called I. Maybe in citation or something. Yeah. Yeah. So what happens here for the PQH here? I mean, like the PQH here, you also have can have a PQH here as well. So for the PQH here, the H can be loaded right into the lumen. So you can see here, if you go this way, the PQH that has been formed here, let me correct this. You can see here, PQH, plastohydroquinone, can be formed at 2 places here. Number one, over here, just before the entrance into the cytochrome B6F, over here. PQB, after PQB is PQH2, plastoquinone. Or it can be formed at the top of cytochrome B6F. Okay, if it's formed due to the Z scheme, the moment it gives off the electron to the Q0 site of cytochrome B6F, the hydrogen is released to the lumen. Why 2H? Because That's the formula of plastohydroquinone, PQH2. So it removes 2, it PQH2, it removes 2 times hydrogen proton. So what happened to the 2 electron? 2 electron, one go up here, one go down here. That's how. That's how you got your 2 electron and 2 hydrogen. Okay? Because remember now you are dealing with hydrogen proton, not hydrogen atom. Yeah. So the cycle continue again, and you can re-obtain your plastohydroquinone. And this plastohydroquinone is not originating from PS2, okay? Not from this pathway, no. This is from the recycling of the cytochrome. Then it can comes back and continue the cycle over and over again. Okay, and plastocyanin. Plastocyanin, I think among all of these protein, I think plastocyanin is the most water soluble and the smallest. I think in the book it says somewhere. Does it say here? It should be very small. Yeah. Oh, by the way, all of this is called the Q cycle. All of this. You see? Go down, go up, go down back. This entire of here is called Q cycle. Specially to describe electron movement within the cytochrome B6F complex. And what is cytochrome B6F? An integral protein. Right. Okay, and finally, after plastocyanin, now we are coming back to the Z scheme. Plastocyanin will pass this to the P700. How do you say this? Ground state P700. Please don't forget that it will get excited. Boom. It get excited, it will pass to PC plastocyanin, then P700, then whoops, P700 star. Right. Then actually I should make it higher. Tak cukup ruang. Star, and then it will start to transfer it to A0, A1, iron sulfur X, then it will go to iron sulfur A, that will goes to iron sulfur B, that will goes to ferridoxin, then it will go to flavoprotein ferrodoxin apa benda tak, NADP reductase, FNR. Then FNR, it goes to NADP plus, eventually it become NADPH. Yep. So it's quite straightforward for Photosystem I. Very, very straightforward for Photosystem I. So all of these, you need to understand, they are all iron sulfur protein. All of them. And sometimes they contain copper as well. Yeah. So that's why you need to give iron to your plants. You know, some old people, they like to put, nails or something into the flower pot. Yeah. So iron deficiency, you can see on the plant, it starts, you have given enough nitrogen, but it still look yellowing. Iron and sulfur. Okay. So iron and sulfur is very crucial to reconstitute the broken photosystem I. Okay. Without iron, without sulfur, none of this would have taken place. No electron can move about, right? Okay, yeah. Oh, I think I was correct earlier. A1 plus Thylakoid. That's the name. Thylakoid. Almost confused with botanical terms. Phylloclade. That's in cactus or something. Right. Okay. So this is the model molecule for this photosystem I protein complex. And again, it is a complex, meaning that it has not only the pigments, but also the protein. You see, not only one type of chlorophyll. This chlorophyll here is present in various color. You got the green chlorophyll, purple chlorophyll, red chlorophyll, blue chlorophyll, meaning that there's different shades of chlorophyll. Okay, that's why when you go to the forest, we got a tropical forest, right? There's so many green here. Petronas green, katak pisang green, nasi lemak daun green, apa? Bendera pas kind of green. So many different shades of green. Why? There are many shades depending on the species, of course. Okay? Right. Kau nampak apa? Terima je. Okay. Right. This is just an enlarged picture of it. Right. Okay. So this is the sequence of it. This thing, I forgot to mention. This thing, Fe, SX until AB, iron sulfur XAB, collectively we call it iron center, I think. Is it center? Iron ferrum center. Iron ferrum center, iron sulfur center. This thing, collectively, come together, 3 of them together, we call them as iron sulfur center. Okay, and also then they will transfer the electron to ferridoxin, soluble iron sulfur protein. Most of these are actually soluble. Okay, they are soluble, right? And then they will eventually oxidize the NADP to become NADPH. What is NADPH? Is it energy molecule? Tak sampai 3 hari. What's an NADP? What's the full name of NADP? Need to correct this as well. Sebelum semua orang dapat ilmu sesat. Correct your pronunciation. Apa ni? NADP plus. How do you say it in words? N for what? Kotak. Nicotinamide, adenine, dinucleotide, phosphate, oxidized. Meaning that it has hole in it. That's why I got the plus sign. It's positive. However, the moment it has received a hydrogen here, how do you call it? There's a hydrogen here. Reduced nicotinamide, adenine, dinucleotide, phosphate. Put the word reduced at the front. The moment you see the compound or substance has been added with hydrogen, the hydrogen automatically put the words reduced in front. Nothing to signify it is reduced in terms of quality, it becomes sadder, it becomes uglier. Nothing to do with that. Reduced here because of this formula here. RIG, rig, meaning reduction is gaining electron. It gains electron from who? From hydrogen, because hydrogen atom got 1 electron and 1 proton. Okay, as simple as that. Right, and finally, I think this is the final part of it for 9.6. With this knowledge, what you can do? You can work with herbicide. Anybody want to work with Syngenta? Syngenta, Bayer. You see, this herbicide, weedicide company, they know some compound like DCMU. What's a DCMU? Dichlorophenyl dimethylurea. Here go. I know you to urea or diuron. Is it diuron? Yes, diuron. And also thylakoid. What's this? Be here. Zaskim. So when you talk about DCMU, the diuron, it will cut the transfer of electron in between plastoquinone. Specifically here, it doesn't have to disturb any other thing, because that's why herbicide, weedicide can be very cheap, because they just need one active ingredient, and this active ingredient cut this and the entire thing will collapse. Other example is paraquat. Paraquat interfere with the reduction of this NADP plus. Okay, so the electron needs to move this way as well. Okay, but it is interrupted by paraquat. Okay, so with this specific method, you don't kill the weeds. You spray it, suddenly it's gone. No. After 3 days, you will start to see that it's wilting, it's yellowing, and then it's just die off. But for those animals that get sprayed with this, not necessarily they are affected, because this might not be present in the animal. This thing, ferridoxin and so on. I don't know. Maybe we got it. I think paraquat killed this. We got this as well. This we don't have, I think, because we got ubiquinone, not plastoquinone. We have it, but our human form. Okay, plastoquinone, plastohydroquinone in plants, in chloroplasts. We have it in our mitochondria. Ubiquinone or ubidecarinone. I think you have seen it in the pharmacy. It's called coenzyme Q10. Eat me, you will stay young forever. Try lah makan. Okay, all right, yay. And that's the summary. Exercise. Bye. Okay. Have you received your quizzes and so on? Take this. No group work. Sebab korang tak ramai kan? Please do the exercise. Whatever that you have answered here, I don't want simply an answer. You need to prove me where is it. Maybe you want to photostat this, cut this, paste next to it, or you want to direct me, this is the answer. Use your creativity to validate your answer. You can do the collage next to it. You want to do the hologram next to it. Anything. I don't want simply you answer this. What are the product of Thylakoid reactions? Betul kau jawab sugar. Why? Grandma said 10 years ago. So aku nak pergi panggil. Masalah dia dalam lubang sekarang. So give me something that can validate. From what I think it is, the notes should be enough. If you go to your folder, the books menatang ni dah scan, ada dalam folder tu. So you can use that as well if you don't like this. The book pages. I don't bring the book. It's too heavy. Physio ni, developmental physiology. The latest released in July. That's the latest. Yeah, courtesy Amazon, give me $20 coupon. Sebenarnya dia nak suruh aku join Prime tu, tak pasal. But I still took the coupon to get a discount. Jahat kan? Okay, please do this. So, remember I told you, this class default is A. This sama ada kau rajin kau malas saja. Okay, if this is enough, maybe this can be your first test. This is only one topic, and then we have another topic. Yeah, and I think in the folder as well, I don't remember whether I've put it or not. There's a summary as well. Yep, summary like this to help you with. This is summary for this lecture slide. I have this, the summary for the entire chapter 9. What keywords that you need to know. Do not move until you know these keywords. Okay, that's how you know whether it is enough or sufficient or insufficient in terms of your lesson. Right? All right. Okay. Senang kan soalan? So 9.1, you should be able to answer up to this point, which is 9.6. Yeah, 9.6. Boleh tiru? Boleh. But I still need to know where you know the answer. Show me where. Show me where. Right. Oh, dah senang. Asal buat susah lagi? 9.6, number 3. In the reaction centers, the first photochemical reaction involves the transfer of an excited electron on the chlorophyll to an acceptor molecules. This process forms apa? Reaction center, first photochemical reaction involves transfer of excited electron on the chlorophyll to acceptor molecule. This process form what? Proton gradient, charge separated state, protein complex, or CG evolving complex. Apa? It gets excited, what happens now? Ni benda ni kat sini. Dah kena padam lah. Prove to me where? Fine. Mana kat nota tu? I want you can use scissors kau nak gunting, tap, and then put next to it. Atau, I do not know. Because the soft copy of this ada dalam folder as well. So you can print. Prove to me that's the answer. Tulis. That's not showing me. I still need to know where it came from. Show me where the answer came from. That's the only way can ensure that you are actually reading the notes. Okay? All right? Sometimes the question can be two answers because you can see that I kind of repeating things from the last week, right? So maybe it's from the notes from last week. Doesn't matter. Correct things, still correct. Anyway, right? It's pretty much like when you're ugly, you're still ugly anyway. Nothing much can happen. You can put all the 3 kilometers of makeup, still not gonna help. Alright, I think that's all. Cepat nanti jam. I got outstation to go.